In this tutorial we'll have a quick look at Mendels Laws. There are two key laws that we need to focus on. One is the law of segregation and the other one is the law of independent assortment. In the law of segregation, this is what is said. Every person carries two copies of a particular allele. So they have a homologous pair of chromosomes. And on those chromosomes they carry two copies of each allele of a gene. Once you've studied meiosis, this law will make a lot of sense. But through meiosis, each member of a homologous pair, so each chromosome in the homologous pair, separates or segregates. That's where the word comes from, separates or segregates. That means is split apart into separate gametes. So this chromosome here will end up in this gamete, and this chromosome here will end up in a different gamete. So each chromosome in a homologous pair cannot end up in the same gamete under normal circumstances. They are separated during meiosis into separate gametes. So the genes that were on this chromosome will end up in half the gametes, and the genes that were on this chromosome will end up in the other half of the gametes that are produced. Mendels' second law, the law of independent assortment, is shown in this diagram here. So let's have a look at what it's saying. We're looking here at a cell that in this case just has two pairs of homologous chromosomes. So there's one pair of homologous chromosomes. The blue, as it says over here, is the maternal set. The red, is the paternal set. The red is the maternal set. So as we enter into meiosis, we have our homologous pairs lined up on the spindle fibre. So we have one pair here and another pair here. Now, in meiosis, the way the maternal and paternal chromosomes line up on a spindle fibre is completely random. It's quite possible to have the blue on the left here and the red on the right, and the second pair could be blue on the left and red on the right, or quite alternatively you could have blue on the left and red on the right, or you could have red on the left and blue on the right. And this applies to however many number of pairs of chromosomes the organism happens to have. We would have 23 pairs lined up for meiosis. So shown here is two different possibilities for the way those two different homologous pairs could line up on the spindle fibre. Now the law of independent assortment, I'm just going to clear away all the ink there for a minute. The law of independent assortment says that the genes found on one homologous pair of chromosomes assort quite independently of whatever happens to the genes on another homologous pair. So the way the red and the blue line up in this pair is utterly independent of the way the red and the blue line up in this pair. It's a completely random process as to whether the red or the blue, that is the maternal or paternal, line up left or right. Of course, the way they line up, the way they arrange themselves on the spindle fibres, will determine how they separate during meiosis. So if you had all the blue down the left and the red on the right as shown here, then the blue homologous chromosomes would end up in one gamete, the red in another, and eventually you'd have half the gametes with blue and the other half with red. However, if they lined up as they have over here, then you end up with half the gametes with a red and a blue here, and the other half of the gametes with this red and blue. So you end up with a different combination of genes or alleles in the daughter cells, that is the gametes produced. Now imagine having 23 pairs of chromosomes and the number of different possibilities that you could have. So this is what they mean by the law of independent assortment. I'll just clear the ink again for a moment. I'll just restate it. What happens to one homologous pair of chromosomes, the way they line up down the equator during meiosis, is completely independent to the way any other pair may line up. And this will determine their assortment, that is their arrangement during meiosis. So this contributes to the mixing up of alleles during meiosis in many, many, many different ways. And all that contributes to variation or variety in the offspring.